in the context of thinking about endpoints for clinical trials, the point that I emphasized in my lecture is that the current standard is clinical acute rejection. The reality, as I pointed out in the talk, is that that's about 10 to 15% perhaps at the most today. The problem with that is that then one powers clinical trials to a year or two at least, 1,000 patients, which means practically 50 or so different centers across the United States and possibly Canada and other areas in North and South America. That's really hugely expensive work for the pharmaceutical companies. And it's been a growing disincentive for pharmaceutical companies to invest in developing new immunosuppressive drugs. And we, as a field, and our patients families lose when that happens. I mean, I'm happy they've switched their attention to diseases that are equally deserving, perhaps, like psoriasis and multiple sclerosis and inflammatory bowel disease, but I'm a transplanter and I'm loyal to the best interest of our field. So how do I address that issue? Well, one point I made, a very critical point, is that the rate of subclinical acute rejection is equally high. In fact, it's probably higher. It's as high as 20 and 23%. So if you add up clinical acute rejection and subclinical acute rejection, now you have an endpoint of about 30, 35%. And that's the kind of thing that would reduce significantly the number of patients and the time it requires to do these studies. So that'd be a huge thing to remove. It's a huge disincentive that could then become an incentive to do these trials on the part of pharma. So that's one suggestion I have. The question would be, how would one document subacute rejection in order to accomplish that, given that the definition of subacute rejection is that you have normal renal function and you're clueless about having subacute rejection. So that takes me to the second piece, and that is the potential value of molecular diagnostics in doing so. And that then goes to the work that we've been doing for over a decade now in the lab in development of genomic tools, molecular tools, if you will, for diagnosis of rejection and the efficacy of immunosuppression in blood and in biopsies. So I would suggest that now we can actually deliver several things to clinicians and pharmaceutical companies. So the first we could do is draw serial bloods on patients. So you might think of a trial where every month, up to 6 months, and then maybe every other month till the end of the first year, one would routinely draw a tube of blood. And then you would do gene expression profiling on that using either arrays, which can be done really readily. We have instruments now in my laboratory that can do about 250 arrays, 300 arrays a week, all in an automated mode. So this is something very well, it's ready to be introduced in a clinical workflow. So that's really important to emphasize. The second technology one could use is what we call next generation sequencing or deep sequencing. And again, with multiplexing and targeted deep sequencing, we can handle hundreds of samples in relatively short periods of time, and they can be automated up front. So the important point is that sometimes scientists are talking about, yeah, let's do this or do that, and they're very impractical. Yeah, sure, I can do 10 patients so a year, that kind of thing. And it costs $300 million, but it's fantastic, and I discovered all this, and that's not the case, and that's what I'm trying to emphasize right now. These are really ready for clinical implementation, and now the question is really getting it done. So that's the next step. A second possibility is to create protocol biopsies. So, for example, in my center at the Scripps Clinic and Scripps Research Institute, we do protocol biopsies on everyone at 2, 6, 12, and 24 months. So a protocol, or what we call a serial monitoring biopsy, is something that we've integrated into our normal standard of practice. It's part of the workflow in our transplant program. Done everything as an outpatient. We can do the work now profiling these biopsies, and these biopsies can be profiled on paraffin-embedded sections. So we don't need, as we used to, we don't need an extra tissue core put in RNA later or an equivalent preservation solution. So people can just do a biopsy the way they normally do that everywhere in the country or the world, and then we can just cut a few sections from the block and profile it. So we could do biopsies, for example, in patients in the first year at 3, 6, and 12 months. So that's 3 biopsies. And with that, you would pick up 20 to 25% of the subacute rejections. One of the ironies here, and I pointed that out in my lecture, is if the incidence of subacute rejection is 20, 25%, and you're only doing 3 days in a 365-day year, what's that telling you of what's going on in the other 362 days of the year? So the only way you can really get your head around that, because otherwise it really doesn't make sense, the only way you can really get your head around that is that once patients are transplanted, rejection is present. And once rejection is present, it's all about the efficacy of your immunosuppression. So in my view, there's 2 kinds of patients that we deal with. The first patient, you get the immunosuppression correct from the beginning. That may be because the patient has no immunological memory. I'll get back to that in a minute in another question. It may be because the patient's genetics are particularly good. It may be because the match with their donor is particularly good, and any combination thereof. In either case, the situation is those patients are going to do well. If we look at 1-year results of these kind of serial monitoring biopsies and the histology and the molecular profiles, we know that that group of patients that does well is probably 65–70% of our patients, which is nice. It leaves us 30% of our patients in the first year, 30 patients out of every 100 that are not doing so well. And those patients are probably having subacute rejection, ineffective immunosuppression, and a background of constant immune activation for the entire year. And that's a little sobering. So now where do you go? So if we can do serial blood monitoring, and we can detect the presence of subacute rejection by gene expression profiling. We can do that on arrays. We can do that by next generation sequencing, and these technologies are all available. We could do biopsies, and we could do a combination thereof. And these would then create efficient surrogate markers for pharmaceutical companies to develop trials that one could do very quickly. Now there's another interesting flip on this. So if we biopsy patients at 1 year, another point I made in the lecture is that we see a whole lot of chronic rejection, this interstitial fibrosis and tubular atrophy, or IFTA. And we recently published in the American Journal of Transplantation, again, which I reviewed in my lecture, our experience in showing that if you look at patients with IFTA, they, by molecular profiling and mapping of these molecular changes to known functional immune pathways, we discovered that they basically have rejection. So now we're talking about this 25 or 30% of patients at the end of the first year who probably have been immune activated for the entire year, or most of it, and they now have increasing evidence of tissue injury because they're developing significant BAMF-grade, biopsy-proven interstitial fibrosis and tubular atrophy with an active immune response. We call that chronic rejection. And now one could think of a new kind of trial. So that trial would be, you basically don't do anything for a year. And at the end of 1 year, you biopsy everybody. So now we're not talking about serial blood monitoring, and we're not talking about serial monitoring biopsies. We're only talking about one biopsy on everybody at 1 year. And if you did that, one would expect to see that 70 patients out of every 100 would be fine. Leave them alone. The 30 patients, approximately 25 or 30, that have evidence now of interstitial fibrosis, tubular atrophy, and immune activation by molecular profiling of the biopsy, if one profiled these patients at 1 year using these protocol or serial monitoring biopsies, you detect molecular activation by the profiling, you detect histological evidence of interstitial fibrosis and tubular atrophy. And if you fulfill those two criteria, then you can initiate an intensification of immunosuppression. The other 70 of the 100 patients who have a normal biopsy and no immune activation should do well. And so now we're intensifying immunosuppression in just the patients that need it. What makes this a potentially attractive trial design for pharma and biopharmaceutical companies to develop new drugs is that, number one, we have blood tests that match the biopsy tests. So you do the biopsy at 1 year, you select your 30 out of every 100 patients that should be the target of your new drug or your new biologic. And you now have a blood test, so you measure that blood test for a couple months. Frankly, if the blood test doesn't normalize, if you don't get blood evidence of going from immune activation, subacute rejection, or subacute chronic rejection to normal immune quiescence again, or efficient immunosuppression, if you don't, then your drug's failing. And you can know that in 6 to 12 months. Now you suddenly have a very doable trial. You're targeting only those patients who potentially could benefit from your drug. The numbers of patients required for such a trial would be dramatically reduced, and the time frame for doing the trial would be dramatically reduced. That would remove a huge number of the disincentives I was talking about before for doing trials, and could energize a new generation of emphasis on studying these drugs in transplant patients again. And that would be very exciting for our field. So that would be my aspirations for molecular profiling and diagnostics in this case. Immunological memory is a really interesting problem for immunology. So when we think about a classic immune response, we think about naive T cells typically, that are coming out of the thymus after an early selection process, circulate in the blood. At some moment, they circulate into a newly transplanted kidney or liver, lung, heart, small bowel, whatever you transplanted. And they go, oh, voila, there's a foreign antigen here I need to activate. And one then goes through this sort of canonical, classic immune pathway of CD4 T cell activation and antigen presentation and co-stimulation, all of which I talked about in my lecture, and is well known and documented. There's two issues. First of all, patients don't appear for a transplant out of the blue. They weren't spontaneously generated yesterday. So they're human beings with often complex immunological histories. The women, for example, may have had multiple pregnancies. At the time of pregnancy, the women are transfused briefly with paternal antigens. Well, maternal as well, but they obviously are not going to make a response to themselves. But they'll have memory against paternal antigens. Now, let's say you come along with a living related donor that has the same paternal antigens, you now have memory. And so the concept that you're really dealing post-transplant with a purely naive immune response is actually not true. As we grow through adulthood, we're also exposed to other antigens. We may get blood transfusions that expose us to antigens. We know there's a bunch of microbial antigens that we're constantly exposed to that shape the immune repertoire. So to think of a patient and the patient's immunological responses after a transplant as purely naive T cells is, if you will, naive. I mean, you basically have memory, and it's there. And the second part of memory that's really important to remember is that you have a transplant for years. So sure, let's just pretend for a minute, which isn't, I just got done saying isn't true, but let's just pretend for a minute that everything was a naive response. Well, what about a week later? now you have memory. 2 weeks later, you have memory. And the naive cells keep coming out of the thymus, and each time they come out of the thymus, when they circulate through the transplanted organ, they go, voila, there's a foreign thing here, and they will develop again into memory. So by the time you get to 8 weeks, 12 weeks, 3 months, 6 months, you're now, the future of the graft is not being determined any longer purely by naive T cells and their reaction. So you have now to think about it in more complex terms. So the outcome of grafts and the challenge to deliver effective immunosuppression is now more complicated. It has to be, you want to inhibit naive T cell activation, because that's occurring. Even if you have memory, let's not forget that you have these naive T cells coming out of your thymus every day, and they're going to activate. So you need to keep them under control. But you also have to deal with these memory cells, which will then accumulate with time. So how do you do that? The problem has been that most of the models that have been used to develop the drugs we use today are based on these, you basically take this mouse who has no immunological memory. These are actually all really young mice too. So we're talking about modeling immunosuppression it's all based on naive, it's all happening in a really short time frame. And we're talking about days to weeks. And it's not a very perfect model for what's really going on in human beings. So that a lot of our drugs are very effective at inhibiting naive T cell immune activation and very poor at dealing with memory. So if you come into the transplant with memory, and I gave you reasons for why most of our patients are coming in with memory, and inevitably you develop memory within weeks anyway, even if you could possibly argue that everything was naive. And so we studied the different immunosuppressive drugs, and we have some really compelling data that the calcineurin inhibitors are particularly effective at inhibiting naive T cell activation, but actually not as effective on memory. In contrast, rapamycin and everolimus, the sirolimus-based drugs, the mTOR inhibitors, they are more effective at inhibiting memory than the calcineurin inhibitors. So interestingly, there was a time in which we thought that we would either use the mTOR inhibitors to avoid the effects of the calcineurin inhibitors. That didn't play out. You know, despite a small blip in use, uptake of use of the mTOR inhibitors, about within the last 10 years, it's really gone away again, and 90+% of American transplants, North American transplants, are done with FK506, the calcineurin inhibitor classes. So how does that really play? Well, it works short term because FK506 or cyclosporin, it really, you know, both calcineurin inhibitors work, as I just said, very well against the naive immune response. So you might do really well for a couple months, but then if you go back to my discussions of a moment ago, that our evidence is that once the patient gets out a month or two, for the rest of the year, for the rest of their transplant's life, these patients have ineffective immunosuppression in 30% of the cases. You have to ask yourself where the hole then is in the effective immunosuppression, and that might be the fact that maybe what we really ought to be doing is starting off on a calcineurin inhibitor like FK506, playing out the naive effect. And then within a couple months, introduce some mTOR inhibition so that you now come up with combinations of a calcineurin and an mTOR inhibitor. And that might then pick up your memory that's now accumulating in time, as I was talking about a moment ago. And that might provide much more effective immunosuppression than our current regimens without changing anything.